All right. Hi, everybody. I hope that you are all doing well. So you should have all gotten an email from me earlier today with your graded discussion questions from the last two discussion papers. Those grades are also posted on Moodle. And I have the exams for you as well that I can give you back at the end of class. And those are also all updated already on Moodle. Remember that the assignment with regard to the Bureau paper is due tomorrow. So you want to make sure that you take a look at that paper and answer those questions. So I think that those are all of the general things I had to mention. I did send you guys a thing about the talk, the RISE talk that is today that I'm pretty excited about. And there is an opportunity to meet with the scientists earlier in the afternoon if you are interested in doing so. So I sent you guys emails about all of those things. Yep, Miles. Tomorrow is not Friday. It's due on Friday. In my brain, tomorrow was Friday, but it's due on. It's Friday. Yeah, Nadia.
For the talk on the RISE 4, is there two different times for two different things? Like I saw one at 3 and then one at 5.
You can meet the people from 3 to 4. The actual talk is 4:30 to 5:30.
Okay.
Sorry,
Correct.
That
is exactly correct. Thank you.
Is it like a presentation at 4:30?
Yeah, it's a talk, and it should be a pretty cool talk. I mean, the drug is really cool, so.
Right.
And it's being zoomed.
It is also being zoomed.
Oh,
So
it is?
yes.
Okay.
So.
I have another Zoom that I have.
So that is the story with all that. So today I'm going to start talking about some details regarding immune memory responses. I also have some stuff here about NK cells, and today's topic on the schedule is NK cells. I know myself in immune memory, so I have a guess about what percentage of this time I'm going to spend on immune memory and how much of it the NK cells are going to get pushed to Friday, which is not tomorrow, but we'll see how it actually goes. So right now we're going to say I'm talking about both today, but we'll see. So we have started thinking a little bit about this idea of memory cells in that they are sort of one outcome of what we might see after activation of our T cells and our B cells. So the majority of the activated cells will die through this contraction phase, but some number will be around afterwards, which will be the memory cells. And the number of memory cells specific for any particular antigen will be higher than the number of naive cells for that antigen. As this image shows here, memory cells are sort of this other potential outcome from our naive cell getting activated. There is a bit of debate in the field as to whether the cells sort of decide at activation, I'm going to be an effector cell or I'm going to be a memory cell, or if some effectors turn into memory cells. I'll show you a little bit of the information that I find compelling on which of those two things happens, but the jury's still a bit out on this. And there are a lot of differences between effector cells and memory cells. One of the biggest differences is shown here in that the effector cells are short-lived. So these cells are highly, highly, highly specialized to do some type of function, but then they're going to undergo apoptosis pretty soon thereafter. While the memory cells are specifically long-lived types of cells. And we talked a little bit previously about how long-lived means. And I told you about the data that are on this slide, where we see that the antibody response doesn't necessarily decline super quickly. T cell memory maybe has a half-life of 8 to 15 years, but we can see memory T cells out to 85 years post-vaccination in older individuals. And in mouse models, we can see memory cells that are living for 3 times the age of the mouse. And the reason why they got to that number is because that's when they stopped the experiment, not because the memory cells weren't still living. So it's probably potentially longer than that. So these memory cells are going to live for a very long period of time. Just like when we talked about naive versus effector cells, we can also see different characteristic proteins that can distinguish naive versus effector versus memory cells. So you can see that there are some proteins that are not really present so much on naive cells. So sort of cells that have not yet been activated, but then as soon as the cell gets activated and is either effector or memory, we start to see more of that protein. Some, like CD69, are really only on the effector cells. There are also some that are unique to memory cells. The memory cells tend to have a lot of proteins that are involved in cell survival. The memory cells also tend to have a lot of different types of cytokine receptors. So one of the key proteins we often talk about on memory cells is called CD127. CD127 is actually part of the IL-7 receptor. And so CD127 is allowing memory cells to get a lot of that IL-7 survival cytokine to help survive for a long time. The memory cells have a lot of BCL2. We also can see differences in terms of trafficking in memory cells. So remember that the naive cells are generally trafficking to the secondary lymphoid organs using things like CD62L or L-selectin and CCR7. And remember that our effector T cells or our effector cells stop going to secondary lymphoid organs. We don't want to kill the lymph node. Instead, we want to have those T cells go to some other kind of tissue to deal with the microbe. So those cells will lose L-selectin. They might lose CCR7, so they'll gain some other trafficking molecules. It'll just vary in terms of where we're trafficking. Memory cells tend to gain back the ability to go to the lymph node, to go to the secondary lymphoid organs, both for looking for different microbes and for picking up some cytokines. So we see a big change in trafficking. One other aspect of this aspect of sort of thinking about immune memory is that we have realized that all memory cells are probably not homogeneous. They're actually different types of memory cells. Just like we see different types of helper T cells or different types of whatever type of cell. So there's probably not just one kind of memory cell. There are probably multiple kinds. They might differ in terms of things like where in the body they are spending their time, exactly how long they live, exactly how quick they are to trigger, things like that. And sort of separating out some of those subsets and figuring out exactly what's going on with some of those different memory types of cells. It is something that is still going on in the field. So sometimes when you read different papers, they'll describe their memory cells somewhat differently because they are sort of thinking about their memory cell subset a little bit different ways. On the right-hand side, you can see an example from your textbook where they talk about 2 subsets of memory cells, central memory and effector memory cells. So the moral of the story is that memory cells are a heterogeneous group, not a homogeneous group. One other really big difference is that with our naive T cell, if we look at the DNA that encodes a lot of our cytokines, that encodes our granzyme B, that encodes our perforin, all of the important genes for different effector functions, that chromatin is closed chromatin, and you can see an example of closed chromatin here. Basically what you can think about is that chromatin is really tightly wrapped around histones. So it is hard for transcription factors to access and get in there and lead to transcription. And so if we want to do transcription of interferon gamma or of granzyme B or whatever effector function type of thing we need, that cell, that naive cell, needs a lot of activation because it needs to do a lot of work to unwrap this chromatin and start doing transcription. So we need a lot of signaling to get our transcription in those naive cells. That also is related to sort of that need for signal 1 and signal 2. With memory cells, the chromatin is way more open, and so it's much easier to trigger these cells to start transcribing any important effector function gene. We generally will have a lot of our signaling proteins pre-made in these cells so that these cells are going to be able to signal right away, much faster than the naive cells. They're going to make a lot more cytokines so that they can survive a long period of time. They also, don't need things like CD28 to get a signal 2 anymore. So we've got some big differences in these cells. All right, so I'm just looking at the order of how I have some things to talk them through. So I've shown you this previously, and we're going to see some more examples of this that I'm going to draw out on the board a little bit later. But this is sort of the general example of the phenomena that we see because of our memory cells. So at the beginning, we start out with a bunch of naive B or T cells that respond to our epitope of interest. We'll actually be exposed to that epitope when we are infected with a microbe. The microbe will reproduce for a while. Eventually, those slow naive cells will get their act together and get activated, and we'll get a large number of them through proliferation, and that will eventually get rid of our microbe. Of course, while that microbe was reproducing, we're going to feel some symptoms. Those symptoms are going to go away after the microbe goes away. And the microbe's going to go away because we had this nice maxed out adaptive immune response that got rid of it. We're going to then see the contraction phase of that adaptive immune response because we don't want to become one giant walking lymph node. And we're going to have this number of cells that are going to persist for some period of time that have changed, that are now our memory cells. And that's going to go on for some amount of time. Later on, if we ever encounter that same microbe again, now we're going to see that those memory cells are able to respond immediately, so we don't have the delay that we had the first time around. Those memory cells are also going to respond much stronger, so the slope here is much steeper. So in fact, these cells are proliferating better. They can actually divide faster. Here, and we get this even higher response of the secondary response. This response is so fast and so high that we're probably going to get rid of that microbe real fast. It might have reproduced a teensy tiny bit, but only a teensy tiny bit, so we're never going to feel symptoms. And so we're going to sort of clear that without symptoms or with much milder symptoms than we would have seen the first time around. And we refer to this as our memory response or our secondary immune response. So one thing I just want to remind you, well, there are a few things that I can remind you of here. One other thing that's interesting to note when we think about memory cells and memory responses is that our memory responses are happening at the same time we're having another innate response. So here's the first time we see our antigen, we make an innate response and as well as an adaptive. And you can see there's basically this time where there's the innate response alone, and then there's the two working together. When we're getting to a memory response, the two are working together from the very start. And we are still going to see an innate response here. It's just we're also seeing an adaptive response happening here as well. And in fact, this innate response is probably going to be helping out this adaptive response. One other thing to note that I don't have here is that if I drew exposure to another antigen happening at this time, we would not see a memory response to that antigen. The memory response is specific to the antigen we have previously seen. We only have a memory response for those cells that have already been activated, have already gone through this whole process and become memory cells. It's not like our whole adaptive immune response has gotten better over time. It's that we have improved in response to this specific microbe. So it's sort of like we have trained some special assassins only against this one target. And if that target's around again, we're going to be better at it. It's not as though we're just generally better at killing stuff. So we can sort of compare and contrast what we see between those primary and secondary immune responses. With the primary response, the number of cells that are specific to our pathogen is relatively small. It is larger when we get to the secondary response. So this is basically just saying in words the fact that this number here is higher than this number here. So we're starting at a higher level with the secondary response than we were with the primary response. You can see that there is this delay before we're getting our response with the primary, and specifically there's a delay with antibody production, there's a delay with effector T cell production. With the secondary response, you actually already have some antibodies that are left sitting around from the previous response. So you have pre-existing antibodies. You already have T cells that are present and can start working automatically. Your B cells have already done class switch. So we already so at the beginning in a primary response, we don't really have class switch. We kind of have our mediocre affinity. After our secondary response, we've done class switch. We already have high affinity, so we already have the good antibodies. The primary response, it takes a lot to activate the cell. In the secondary response, it doesn't take as much to activate the cell. And in the primary response, the innate immune response is going to work alone for a little while before the adaptive response gets out of bed. While in the secondary response, the two innate and adaptive responses are working together. So you can see we've got a bunch of differences here. I'm going to skip over the next slide because I think it goes better a little bit later from now. Because I can, I'm going to say a little bit about this question of how exactly we get from a naive cell to a memory cell, and some of the different hypotheses and different pieces of data that are out there sort of talking about whether our naive cell goes straight to being an effector cell or straight to being a memory cell, or whether it goes naive, then effector, then memory. So I'm just going to mention a little bit about some of the models of how this works. And so you can see that some people think that we have a naive cell that somehow is chosen to become a memory cell, and other naive cells somehow are chosen to become effector cells. So some people think that that's sort of what's going on. Other people think that those memory cells come from effector cells that have lived. One of my absolute favorite papers on this actually shows the data behind this model that's shown over here, which says that when the naive T cell divides, it actually sets up its division so that one daughter cell is close to the APC and one is far away. So you can see this division on this plane right here. And the cell is actually set up so the one that's closer to the APC becomes an effector cell and the one that's farther becomes a memory cell. And so this actually says that the naive cell is sort of the same naive cell, but when it divides, it makes one of each. so that you can make an effector and a memory cell. And the data from this paper, where they used actually microscopy and could track the cells as they divided and where different proteins were at different times, is gorgeous. Yes?
So are those the same model?
So no, this is, yeah, these could be thought of as the same model. At this point, honestly, I think there are probably about 50 models. But the idea is, in some ways, I think that sometimes students get this idea that it's just the memory cells are like the surviving effector cells. That may be true, but it may also be that the cell at the beginning was made into a memory cell, and it was going on a memory cell path even from the beginning. And so I just want you to realize that those cells might have been destined to become memory cells from the very beginning, which is kind of what it's talking about here, that when this cell divides, this daughter is going to be the effector, this daughter is going to be the memory. Yeah?
Other than geographic positionality,
Mm-hmm.
what would the advantages to become either memory cell versus a T cell? What's this choosing process?
So there's all of the stuff that's in the model here about proximity and sort of which proteins are where during the division. The other part of this is the other things that people think about with this are what cytokines do the cells receive, and so maybe you have to get a particular cytokine to learn how to be a memory cell. So only some cells get the memory cell cytokine, there are also some models that it's about signal strength. And so different amount of strength of signal will signal you to become a memory or an effector cell. So there's a lot of debate about which of those things might be going on here. Yeah. And so sort of to get at that, there is in fact this idea that some people think that if we have, sort of a relatively small amount of signal from our naive T cell, then that T cell is more likely to become a memory cell. And in this model, they're thinking of memory cells as being very much like stem cells that are not very differentiated. They're basically like able to divide and divide and divide and divide and divide for a long time and become lots of other things. While as those cells get more and more signals and more differentiated, those cells might become effector cells. And so here you can see that this is a separate model that sort of thinks about this as a signal strength type of thing. How is that cell originally turned on, and what kind, sort of how much ability to make more of itself does that cell have? How much self-renewal like stem cells? So there is this whole model of whether memory versus effector differentiation looks like what's going on with stem cells. No matter what, we know that there are a lot of cytokines involved. Exactly how much of it is total cytokine driven is not clear, and how much of it's just a response to cytokine. But there are absolutely cytokines that are super important for our memory cells. If you look at this image, looking at T cells at different stages of their life, you see the importance of IL-2 really early on. You can also see the importance of a cytokine called IL-15 that we didn't talk about. And you can also see IL-2, sort of all of these things involved in that original growth and survival and proliferation of those T cells. IL-15 is in the same family of cytokines as IL-2. It's part of the common gamma chain cytokine family. So it's sort of like, you got cousins who are both working in this same field. In memory cells, we tend to see IL-15 and IL-7 be really important for allowing those cells to stay as memory cells and to act well as memory cells, as well as the cytokine IL-21, Barker et al. Those cytokines, IL-15, IL-7, and IL-21, are all still part of this same family. So it seems as though many of the members of this family are particularly important in allowing T cells to become memory cells and sort of to support them through all stages of their life. And so we certainly need some memory T cells, need some cytokines to help them live for that very long time that they're going to live. The rest of what I have here talking about memory cells is really about me going through some specific examples of some things. And so I'm going to sort of walk us through 3 different types of examples to talk a little bit about some of the actual details of what happens with different types of memory responses. So just because I skipped over this slide, really what I'm talking about with all of these examples is how a particular immune response that you might have varies over time. And so what we're going to see is we're going to look at kind of some scenarios with some different individuals, and we're going to see that over time, because of some of the details of what goes on with memory responses, we are going to see big differences in the patterns of their immune responses. So if you wanted one take-home message from the responses the examples I'm going to show you, and in particular, one of the examples I'm going to show you, which is the 3rd example, is one that is like, so many immunologists like to talk about it. It's pretty famous in immunology, and most people, like, I think I understood it after I'd really been working in the field for like 20 years. So it's pretty tricky to understand. But since everybody talks about it, I feel like I have to introduce it to you, even if you're going to look at me and be like, what? But the big picture of all of this is that we're going to see that in different individuals, the same infection can result in different outcomes because of some of these differences in protection over time. And so if you were to think about things we've learned this semester, one thing you might think about is that different individuals have different MHC. And so in that way, you could be like, oh, that's one reason why the same microbe could lead to a different disease or different severity in different people, is because of the differences in their MHC. The differences I'm going to tell you about here relating to memory responses are another reason we might see differences in severity. And so that's a big reason why I'm telling you about all of these types of phenomena and ways that protection varies over time. And so we're going to kind of start thinking about this in thinking about an immune response to one particular microbe. I will tell you that some of the details of at least this first example I'm making up a little bit just for like convenience sake. So we're just going to go with it, all right? So what I want you to imagine first is my grandmother, okay? And the reason why we're imagining my grandmother is because my grandmother was born in like 1920 something, okay? Before there were vaccines and things against measles. And so everybody got measles. In fact, there was no measles vaccine until the 1960s. There wasn't sort of frequent uptake of the measles vaccine until a bit after that, okay? So we're going to imagine my grandmother who's born way before this. So we're going to make a graph, and it's going to have amount of adaptive immune response. You could imagine this as B cells, adaptive, I think. Immune response. You could imagine this as B cells. I'm going to imagine it as T cells because I like to imagine T cells, but you could pick whatever you want, okay? And then we're going to have an x-axis, which is going to be time, okay? So let's imagine my grandmother was one of 5 kids. At some point in time, I do not know when, my grandmother got the measles because everybody then got the measles. Measles is caused by infection with a virus, and in a way that will become interesting and important later, measles is the measles virus is a virus that goes away. So basically, after you have measles, your body can clear the virus. It's known as an acute virus, okay? So at some point, my grandmother had the measles. And she started out with no immune response to the measles. It stayed pretty low. And eventually, she made a primary response to measles, okay? So here's her primary response to measles. So let's imagine at some point during here, my grandmother, one of her siblings got measles, okay? So here's where she got measles. I should use the red chalk, red spots, but I won't. Here's where my grandmother got measles. So let's imagine that like here, her brother got measles. Okay? Her brother has never had measles before. What is going to happen with her brother at this point in time? Yeah, Jeff.
Same thing that just happened to your grandmother?
Yeah, he's going to have the primary response. He's going to have the measles. He's going to get sick. He's going to be in bed. She is going to be around. They have a small house. She's going to be around him. She's going to get the virus from him. At that point with the arrow, she gets the exact same virus he has. What's going to happen with her? Yeah, Jazz.
She's going to get a secondary response?
She's not going to get a secondary response yet. What were you going to say, Paige? Oh, okay. Yeah, Emily, what were you going to say?
At that particular point, aren't her effector cells working at peak capacity?
Yeah, at that point in time, she has an amazing set of effector cells working. She might be exposed to the virus, her brother might breathe the virus on her, and her body is just going to get rid of it immediately. The virus is really going to be gone. It's her immune system is not almost going to register it. Her body's not even going to register because those effector cells are going to be so good at this. And so because she's at a different phase of her immune response, she has this protection that he doesn't have. Sometimes we call this situation something called an abortive infection. So basically the microbe tries to get into her, tries to infect her, and her amazing effector cells that are going right there are just going to stop it in its tracks and sort of stop this infection from ever being able to really take hold. Yep, Jess.
So the initial antigen and virus is still, is it?
That's a debate. That's actually a debate. So
Okay.
We'll say, yeah, don't get super stressed about it, okay? It's either very low or gone, okay? All right, so this happens. She's got measles, right? Now, years later, she has a sister who's a lot younger than her. Her sister gets the measles. What's going to happen to my grandma? And so her sister's going to be around in the little house and breathe on her. What's going to happen to my grandmother with regard to the measles? Now she's going to have a secondary response. So now we're going to be here. We don't have effector cells anymore, so we're not going to sort of completely block the virus from coming into the body. But we're going to see a much higher response, much faster response, no delay. We're just going to get this really nice secondary response. And in fact, she's probably not going to have any symptoms of measles, even if the virus got in a little bit. It's not going to ever be enough for her to ever notice she had measles, okay? And we can see the secondary response. I'm drawing it as kind of skinny. It might not be quite that skinny, but we've established by this point in the semester I'm bad at art. So this is what we got. And you can see that it contracts, and it is going to contract with some half-life at some rate, okay? Now, during her life at different points in time, my grandmother babysits random kids. What's going to happen at some point, you think, when she's babysitting random kids in the, 30s or 40s? Yeah, Jazz.
She's going to get measles again?
somehow she's going to be around the measles virus again, right? She's going to see measles virus again from somebody in her community and get measles again. So at some point, you know, she's going to be here. She's going to get measles again, right? And what's going to happen to her now? Sorry, I'm going to write this as my secondary response. Yeah, Jazz?
So I have a question.
Yes, okay. Yeah.
Because that, I don't know if this is a drawing thing.
Yeah.
But
Yeah.
the point that you're currently at is at the same point at the peak of her primary response. Is this indicative?
I sloped it down a little bit, so it's not at that quite at that same peak. Okay? But it doesn't totally matter because these are memory cells and the other ones were effector. So, but that's okay. All right? What's going to happen to her now? She's going to make another response. In fact, that might be too big. This is actually called a tertiary response. So she's going to get a tertiary response. And it's going to go down over time, right? So now my grandmother has my mom. When my mom's a little kid, my mom gets measles. So what's going to happen when my mom gets measles? Yeah, my grandmother's going to get infected with this virus again. She's going to make a quaternary response. And she's going to keep getting these sort of, she's going to keep seeing this virus that she's getting from people in her community, right? And that's going to sort of keep pushing her response really high. So it's entirely possible, probably pretty likely, that at some point, maybe in the 1960s, my grandmother never saw the measles virus again. Okay? That's okay, because she's up here. Yeah. There's a while, but she was at such a crazy high level that she's probably not going to get to a really low immune response and be at a point where she has to worry about the measles very much at all. Okay? So you can see this situation. Now let's contrast this with me, and then we're going to do the same thing again with one other scenario, okay? So now we're going to look at me. I probably should put it I should probably put it at the same level, so like it's easier to compare. Okay, here's me. I got the measles vaccine. Here's my primary response that I got to the measles vaccine. In fact, when I was a child, the measles vaccine was 1 dose. So I got 1 dose of the measles vaccine. Measles vaccine actually got changed to become a 2nd dose, to become 2 doses when I was older. So I did get a 2nd dose of measles vaccine. Okay? Got my 2nd dose of measles vaccine in 1998. I remember it. Have I likely encountered anyone with the measles since 1998? So what's been happening to me since 1998? So in fact, I've been having my immune response wane for all this time. So what I want you to see from all these examples is that memory responses do wane, but if you're getting sort of frequent boosters from your community, and I don't mean boosters like a booster shot, I mean frequent boosters like people in your community are sick and giving you the microbe, you're going to kind of get this effect of pushing up your immune response. And so you're going to see differences in what types of responses you see. We are starting to see that a lot of the things that we used to think the vaccine lasted a really long time, now that we actually are really testing it in a situation where you don't ever see the microbe again, like happened to me, we're realizing, oh, some of those don't last as long. They're now starting to talk about, do we need to give third doses to people? Because now I might come in contact suddenly with someone with the measles because of people in the United States starting to have measles again because they're not vaccinating their children. And maybe if it was my grandmother and she'd gone a while since her vaccine, it wouldn't be a big problem because she'd been getting all these community boosts. She was up such a high level. I haven't been around and I have not seen those measles antigens since I got my measles vaccine. And so, in fact, the CDC keeps having to change recommendations and re-up certain boosters because of these issues. And so what I want you to see from these examples is that your infection history influences what happens when you get infected. So my grandmother had the same virus at all of these arrow times, but she got different outcomes because here she'd had measles before. So her history of having had measles before changes her outcome. So basically, in two different people, if they're infected with the same virus, whether or not they're making a primary versus a secondary, what their infection history is with that microbe changes what the outcome is in terms of severity of that response. Jazz, I saw you had a question.
This is kind of a sideline.
Yeah.
When did the measles vaccine change into like an MMR vaccine, like all three at the same time?
I have a book in my office. I read it. I interviewed the author. Late 60s, not positive. So that's the short answer. It's a great book about it. It's in my office if you want to know lots of details about it. Yes?
Does it matter, so if your grandmother had gotten the measles vaccine, but like not actually the timing-wise,
Mhm.
like it was like further up,
Mm-hmm.
like when it like all of the memory cells are like kind of like declining, would that be a bigger response or is it still the same?
Yeah, so if she got she got a vaccine and got number 5, it would be bigger again. Yes. There's sort of some debate in the field about where and whether it can max out, and that's something that people are arguing about. There's this great case study. There was a guy in Germany, for reasons unknown, who got 273 COVID vaccines. Yeah, he says for personal reasons. My guess is either he was super worried and paranoid, or he had friends who were supposed to get vaccinated and didn't want to, so he was going and getting their vaccine so that they could get a card. I'm guessing it's one of those two things, but I don't know. And he hasn't been said, but he got 273. So like, sometimes people have actually looked, and he has a lovely response. It doesn't seem like he seems to have no ill effects from having 273 COVID vaccines. But yeah, so like, yes, there's debate about how high it can go, but it's sort of hard to exactly know the real answers. I have a podcast episode about that paper. I could find that paper if you want to see it. Yes?
So is the idea that like every time that your grandmother's memory cells like had the opportunity to do something, they like proliferated, and that's why it doesn't like go down?
Yeah. So basically, she sort of got another stimulation of those memory responses, and so they again you get the response to a secondary and then a tertiary and then a quaternary, where you get even better and better memory responses and stronger memory cells, and sort of you're improving the response every single time that you see that antigen over time. So if you've seen it a bunch of times, you're going to make a really, 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 really good response.
And like even in quantity, like you probably have more of those.
You absolutely have more in terms of quantity, and so it's going to take longer for them to decline. Yes?
So let's say your grandma had her primary infection when she was 20, and then she had her second when she was 25, third when she was 30, fourth when she was 35. But then she didn't get infected again with measles until she was 75. And so her memory cells would have been depleted by a lot.
Mm-hmm.
And so, is there a chance that her fifth exposure to the antigen could potentially, comorbidities aside, could it be worse than that of somebody who's on their third?
So the answer to that is that that's a really hard question to answer, because you have an extra variable in that experiment, which is that she's old later on. And so your age also matters. And so your ability to make responses also changes as you get elderly. And so it's hard to answer that question because there's also this additional confounding factor of her being elderly. Yeah. So that's one part of it. But I will say that there was a lot of debate, continues to be a lot of debate. Remember when you guys got your first COVID vaccine? Say there was, you could get either the Pfizer or the Moderna. Do you remember one of the big differences between Pfizer and Moderna way back at the very beginning? Okay, so there's something about, what was that? At the very beginning, they both were 2 shots. But one of them was 2 shots 4 weeks apart, and the other was 2 shots 3 weeks apart. And so there's sort of this question of like, does that much spacing matter in terms of where, does it matter if you're here versus here? In terms of, is it mostly effector cells can you go too early for a booster and really just be hitting your effector cells? Do you have to wait long enough for memory cells? When we were talking about this on podcasts, I was like, at the time, I was like, this is weird. Because when I did experiments in mice and I would give them second doses of things, I wanted to make a good memory response, so I waited 8 weeks. So it seems weird to me that we're giving our COVID boosters at 3 or 4 weeks. I was like, I think we'd actually get a better response if we waited longer. Why didn't we wait longer? First of all, like, I don't want to like shade the pharma companies. Why did they go with a short time? Nope. Well, kind of herd immunity. They wanted to get everybody's immune response as high as quick as possible. They didn't want to have everybody like waiting around with their low. They wanted to get you high as possible. And maybe you were not getting as high of a secondary response as you could have if you waited longer, but you were getting a nice protective secondary response, and they didn't want you to wait. They wanted everybody super protected as early as possible. And so, yeah, it makes sense that they were giving the boost really early, but I was kind of like, is that the best time for boosting? And there are some papers now, like much later, where people are actually doing the test of how long to wait for the boost. The reason why you generally need a third COVID shot is because the second one was too close to the first one. And we now realize the big reason why we're giving the third one is because we need a little bit more spacing on those timings. And so that's the other thing that relates to your question, Jazz, is there may be some question of them being too close, of it being potentially too close before you have good memory cells. And that's something that I think people are just starting to like really work out now since the mRNA vaccines with COVID. Yep, Miles. Okay.
Was it the COVID vaccine that we discussed where like 3 weeks after it you can't even like be infected?
Yeah, it's for the first 7 weeks, yeah.
So you can't even be infected by it, so you can't be diseased by it, but then after that time you can be infected, but you likely won't.
Yeah.
So if you were exposed to COVID within
Mhm.
was it 7 weeks, 8 weeks?
7, yeah.
Then you likely wouldn't spread it to anyone else because you're not even infected.
Right.
But if you got it like 15 weeks after, does that mean that you can still infect other people?
That is an exceptionally difficult experiment to do. The answer is yes, we think so, but it is hard to experimentally totally verify that. Is the short answer. I could go on and on about the experiments people have tried to do, but I got enough I can go on and on about memory, so we'll go with this right now. All right, so I want to change up this scenario a little bit. So that's why I left my 2 drawings on the board. So now we're going to look at this drawing on, and basically what we're going to do with these 2 drawings is we're going to have the exact same scenario, but we're going to change the microbe. So instead of having an acute infection, we are going to have something called a chronic or persistent infection. So there are some microbes that once they are in your body are there for the rest of your life. Your immune system cannot fully get rid of them. It can sort of keep them in check, can keep them down, but it can never get them to 0. The most famous examples of this are herpes viruses. If you ask a virologist, one joke in virology is that herpes, unlike love, is forever. Any viruses in the herpes group are in your body forever. HIV is also a persistent virus. So basically, once you have HIV, you have it forever. So we're going to look at these exact same scenarios. This one over here on the left now is going to be my mom. The one on the right is still going to be me. And the pathogen is not measles virus anymore. The pathogen is now a virus called varicella zoster virus, which is the virus that is the causative agent of chickenpox. So we're going to now look at the virus that causes chickenpox. And it is a persistent virus. So once you have chickenpox, you have this virus in your body for the rest of your life. Ideally, what happens with this is that the immune system kind of keeps the virus mostly in check. Every so often, the virus can maybe break out of jail. And when the virus breaks out of jail, you have shingles. So this virus is also the causative agent of shingles, and shingles is basically what happens if the virus starts to win for a little while, if the immune system can't keep the virus totally down, okay? So we're going to imagine this scenario. So my mom, at some point in time, had the chickenpox. And so what we're going to think about here is that there's going to be, it's going to be slightly tricky for me to draw, but I'll make it work. There's going to be some level of adaptive immunity that is needed to keep the virus under control, to keep away the shingles. As long as you're over this amount, no shingles. If you get under this amount, you get shingles, okay? Officially in the field, this is called the threshold of protection. And I'm going to draw my threshold of protection. I got to draw it pretty low because I got to be under this point. So, here's the threshold of protection. Yes, I know I'm not drawing a straight line. Again, my art is terrible. There's my threshold of protection, okay? So at some point, my mom got chickenpox. She made a primary response to chickenpox. Then some kid in her kindergarten class who sat next to her got chickenpox. Didn't really matter. She got an abortive infection because she had this nice effector response already happening. At some point later on in her life, some other kid she babysat got chickenpox. She got this boost of chickenpox. Then my mom is a retired elementary school teacher. So at some point, some kid in her class had chickenpox. So she got another response to chickenpox. You can imagine this probably happened more than once of all of these things. It just gets a little hard to draw. Then my mom had me, and I had the chickenpox, 1984. She got the chickenpox. Are you very worried about my mom getting shingles? Why are you not very worried about my mom getting shingles? Yeah, Emily.
Because she's constantly getting exposed to the microbes, so her immune system has the opportunity to get stronger.
Okay, so she was exposed many, many times. She had she made a tertiary, quaternary, she made a huge response. She got a very high memory response. Her memory response was boosted through community boosting many times. And so yeah, her immune response is waning over time, but she's not that likely to get shingles, right? Because she's not that likely to ever have her response decrease to the point where she's below the threshold needed for protection. Now, one variable on this is I'm pretending that she doesn't change in age, that she doesn't also get older, and I know that that is not true, but let's just go with that, okay? So I'm going to give you a couple more scenarios, because like I said, because I can, related to this. So let's first think about me. I got the chicken pox in 1984. To my, maybe, I guess it probably happened that I was still living at home when my sister got the chicken pox. So my sister got the chicken pox. I got the chicken pox from her. To my knowledge, I have never been around someone with the chicken pox since then. So let's say my sister had the chicken pox in 1994. That's about right. Maybe 96. My sister had the chicken pox in 1996. What's been going on with my immune response to varicella zoster virus since 1996? Yeah, Lucas. It's been waning. Are you very worried about me getting shingles? Yeah. My age bracket is the poster children for people who are going to, like anyone who was born around when I am, because basically community spread of this virus went away after we had this virus. People my age are getting shingles. There is a cool paper cool in quotation marks. I don't know if cool is really the right word for this. There are a couple papers that have come out in the past year that have shown pretty well that having this virus trying to reactivate, having this virus testing your immune system, because it lives in the brain, is actually a major contributor to dementia. And so in fact, if you don't have a good immune response to this virus, then you end up with dementia as well. Yeah, David.
Wait, so why doesn't the body self-react to your own shingles virus?
So this is a virology type of question, and so because of the way that the virus is hiding. And so it is hiding in a way that cannot be targeted. Yeah, Paige.
This study, if it's for people around your age, is it early onset?
So this is the issue. So they did an epidemiological study in older individuals.
Okay.
They were not looking at my age group.
Okay.
When the shingles vaccine was first described, I think I'm getting this right, they said that you had to be 70, I think, to get it. Then they said 65. Then they said 60. Then they said 55. Then they said 50. And they're just sitting there at 50. And as a 45-year-old, I'm really annoyed because I don't want dementia. Because the idea is if you get the vaccine, then you're going to bump back up and potentially, get around some of this. I personally have seen no data that would say that would be a problem for a 45-year-old to get vaccinated to protect against dementia, but that is not what the CDC says. Yes?
So, I don't need the shingles vaccine because...
I'm going to come to that in a second.
Oh,
Okay, so then there's this question. So this technically gets into some of our discussion about vaccines, but I was realizing as I was talking about this that it would be interesting, so I'll just go there now. Like I said, I'm not getting to NK cells today. All right, so how many of you guys have had chickenpox? One person, 2 people. All right, 2 people have had chickenpox. What? Okay, all right. No, so sometimes you may not make an appropriate primary response, as we will see when we talk about vaccines. Vaccines are not 100% that every person who's vaccinated is protected, and so potentially that could be what happened in this case. Why did the rest of you not get chickenpox? Because you got a vaccine that was not available when I got chickenpox. It was not available when my sister got chickenpox. Turns out there's 2 versions of the vaccine. There's an old version and a new version. I think you guys are still in the age group that got the old version. I think. I think you guys got the old version and not the new version, but I don't remember the year that the change happened. I will be perfectly honest with you. The old version of the chickenpox vaccine leads to a persistent infection. So you are persistently infected for life with this vaccine virus. And so at least in theory, you are going to be susceptible to shingles when you get older.
Wait, I was going to ask, isn't that a terrible idea?
It's better than dying as a child of chickenpox. It's an excellent question. So in fact, it is so look for live attenuated versus subunit. So basically, the old vaccine leads to a persistent infection. Exactly whether or not it leads to shingles, I think the idea is that it leads to shingles less often than the natural virus, but it's still kind of a little bit like uncertain. The new vaccine doesn't lead to persistent infection. And so you don't have that persistent infection. And so eventually, we're going to eradicate shingles because nobody's going to get latently infected, get this persistent infection with this virus. But right now, we're in this sort of in-between period, whereas people get older, we need to make sure we're giving them shingles vaccines to make sure that their varicella-zoster response is enough to keep this virus in check. So yes, that was the example I was going to tell you that I realized you were going to ask me about. Okay? And again, I have to look to see what the difference is in years for those 2 different vaccines. When we think about persistent infection, there is one other thing that sort of gets a little bit tricky with all the things I've shown you here. This is not the case in the shingles situation, but it is in some others, is that sometimes we also have a situation where we have to worry about the T cells getting exhausted if the antigen never goes away. And so sometimes if we have antigen persist for many, many years, our T cells will go to a state called T cell exhaustion, where they will stop working and may even undergo apoptosis. So one other variable that comes up when we are thinking about a chronic infection is that we sometimes will get our T cells going to T cell exhaustion. The virus is like varicella zoster that's hiding. We don't exhaust the T cells, but that's okay. So I like thinking about chronic infection stuff. So there's one other issue that we have to think about. And this sort of does start to, show you a couple places where I maybe oversimplified some biochemistry earlier on in the semester. Which is that sometimes we do have immune responses that cross-react. So sometimes an immune response to one thing will be able to react with something else. This example is showing you 2 viruses that are very closely related, smallpox virus and cowpox virus. They are both part of the genus orthopoxvirus. And because they're such closely related viruses, they have some proteins that are basically the same. They have some conserved proteins between them. So you can see here's cowpox virus. It's got this green antigen. It's got the red antigen and the blue antigen. Here's smallpox. It also has that same green antigen, that same blue antigen, but it also has a unique antigen that's purple. What we realize is if you get infected with, say, cowpox virus and you make an immune response to cowpox virus, you could make immune responses to the green, the blue, and the red antigen. If you then later are infected with smallpox virus, those conserved antigens will basically result in you having a memory response, even though you've never actually been infected with this virus before. And you will have a memory response to smallpox. In fact, this is how the smallpox vaccine works. This is also how the Mpox vaccine works. We use other non-pathogenic or less pathogenic, less severe viruses in this family that have some conserved proteins, with the idea being that will induce an immune response. Here it's shown as antibodies, but you would also have T cells. So that if you get the actual severe virus, you have this other type of response. And so this is really only true when you have, these closely conserved proteins, these very closely related microbes for the most part. There are a few cases where we found that, like, unexpectedly, based on probability, there are two microbes that end up having the same protein, and it's super weird and no one ever expected it. But usually it's something like this that we see with the smallpox, cowpox situation. This reaction, that's technically the reaction against cowpox that is also fighting smallpox, is called cross-reactivity. So these are cross-reactive immune responses. And this phenomenon that we see here is called cross-protection. So the idea is that after you've had an immune response to smallpox, you are now protected, immune response against cowpox, you are now protected against smallpox. So you've been protected against something that you haven't seen before because of the similarity of antibody epitopes or T cell epitopes and this memory response that we're seeing. And so again, the idea here is that your past immune responses are going to influence what type of response you have. Your infection history is going to influence what kind of response you have. So if someone right now came into this room and released some smallpox, that would be highly problematic, and I don't think that would be a good idea for anyone. I have been vaccinated because of lab work with viruses in this family. So I've been vaccinated, so I have cross protection. It's unlikely that any of you all have. So my past infection is going to give me protection, and I'm going to have a different outcome than the rest of you because of my infection history, my past infection with cowpox. So this can in fact end up leading to sort of complex scenarios when we actually look at what's happening with people over time and their different types of infections. And so I want to walk you through one type of scenario of this. And so this is this one scenario that is really sort of a consequence of these memory responses. If you end up sort of Googling around trying to find out information about what I'm telling you about, the phenomenon that I am telling you about has a few different names in the immunology field, but the one that you hear the most often is original antigenic sin. So the idea that your past or original infections somehow bias your later infections. Your past infections change your later infections. Is sort of this idea of original antigenic sin. There are so many places where people argue and debate about this, and I can totally imagine some questions that you ask that I'm going to be like, yeah, we argue about that. But we're going to just go with this as a very big picture overview. And again, even if you get super confused by this, I want you to realize that the key message here is that your infection history is changing your outcome. So two people infected with the same microbe might have different outcomes because they are because of their previous infection history. So some of the numbers it has here on the bottom of the slide about days in bed, I don't love, okay? So we're going to kind of ignore them. But we're going to talk more about what's going on at the top. And what we're thinking about at the top is infection with a virus that circulates, say, every year. I like to imagine this slide is about influenza. So influenza changes genetically every year. So you can see in the first year, there's a strain that has antigens A, B, C, and D. Then you can see there was some genetic change in the second year, so there's A, B, C, and E. Then third year, there's A, B, E, and F. Then A, G, E, and F. So we can see sort of this genetic change that's happening in this virus over these different years. And so let's imagine first that we have a person. The person's going to be me. We have me, and I get infected with this virus that has A, B, C, and D antigens, okay? When that happens, I'm going to make 4 immune responses. What are those 4 immune responses? Yeah, Jazz? A, B, C, D, and what kind of immune responses are they going to be? Primary. I'm going to make 4 primary responses. A primary to A, a primary to B, a primary to C, a primary to D. You know, one of these kind of things, right? All right, I'm going to be really sick. We don't have to worry about the exact details, but I'm going to be real sick. It's going to be terrible. I'm going to have the flu. The next year, flu undergoes some genetic change. A, B, C, E. Okay? Now, I'm going to get this virus, and Emily's going to get this virus. Emily has never had this virus before, okay? It's her first ever flu infection. What kind of response is Emily going to make in this situation? Yeah? She's going to make the 4 primary responses to A, B, C, and E, right? How is Emily going to feel? Bad. She's going to be sick in bed, right? Okay, what am I going to do? What kind of response am I going to make to this virus? Yeah, Paige. I'm going to make 3 secondary responses to A, B, and C, the thing I've seen before. Might not even make a primary response to E, because we might get rid of that virus so fast, because remember those secondary responses are happening really fast with no delay. I might not even have time to make a primary response, because my secondary responses are going to be so amazing. I got 3 amazing secondary responses. We're going to get rid of this virus real fast. I'm never even going to make that primary response to E. I do not feel sick at all. You ask me if I had the flu, I'll be like, no, I didn't have the flu that year. What are you talking about? I was fine, okay? Third year happens. Influenza has now undergone some genetic change, A, B, E, and F. So you can see the genes in influenza here. Now I get this virus again, having a bad time with flu viruses, and Nadia gets this virus. This is Nadia's first ever flu infection. What happens to Nadia in this case? She makes 4 primary responses. She's really sick, right? The virus, what happens to me? Yeah, Jazz?
You only make 2 responses.
Okay, I make 2 responses, but they're like tertiary responses. They're 2 really, really, really strong, really, really good responses to A and B. Probably don't even make the responses to the other antigens, right? Because we got rid of the virus so fast. Do I feel sick? No, if you ask me, did I get the flu? I'm going to be like, so yeah, it says mediocre. I don't believe it. I don't get sick. I don't feel sick. You ask me if I got sick, I'm going to be like, no. Again, notice last year, Emily and I got the same virus, but we had different outcomes because of our infection history. Here, Nadia and I have the same virus, different outcomes because of our infection history. Okay, now the 4th year, the virus comes around. A, G, E, and F. I get it. Aurora gets it. It's her first ever infection. What happens? Yeah, Jazz.
She's experiencing a primary response, so she's getting all that malaise and sick stuff in bed. But you are on your 4th response, and you are responding to one of the epitopes, and you feel like nothing.
I feel totally fine. I'm making a quaternary response to antigen A. I'm getting rid of that virus so fast. I'm going to do such a good job. I'm not sick. You ask me if I had the flu, I'll be like, no, what are you talking about? I never saw that virus this year. Now the 5th year comes around. The virus is G, E, F, and H. And I get it, and Julie gets it. What's going to happen? Yeah, Jazz.
So here's the gag of it all, right? So you've been making a response with A, B, C, D, and there's been a different change in epitopes each year, but now none of the epitopes on the virus are ones that you are familiar with. So you are experiencing a 5th response to it, but you are experiencing all the symptoms now.
Yeah, so I now am making 4 primary responses. Even though I've actually seen E a bunch of times, my response to E was swamped out or hidden by what I had been infected with before. The fact that I was infected with something else before E kind of like wiped out my E response. My original antigen influenced my response. It was like the original antigen sin. I know that this somehow gets us into Catholicism, but whatever. But so I've actually seen many of these antigens before, and yet I'm going to make a primary response. I'm going to be really sick again. And all of this is because of the way that my past infections were modifying what type of replication I have, what kind of disease I have. And so what I hope that you got from all of this is that past infection history matters a lot and is one of many reasons why we see such variation from person to person. And I will talk about NK cells on Friday. So I'll see you guys then.